In this video demonstration, we will see about a differential bacterial staining technique. The acid-fast staining. This protocol is used to differentiate and to identify the acid-fast bacterial organisms. The differential bacterial staining technique. This is used to differentiate the bacterial organisms. The Gram staining and the acid-fast staining are the differential bacterial staining techniques. The Gram staining differentiate the bacteria into the Gram-positive and the Gram-negative bacteria. In the previous video demonstration, we have discussed about the Gram staining technique. The link is available in the below YouTube description. Next, the acid-fast staining, which differentiate the bacteria into the acid-fast and the non-acid-fast bacteria. The acid-fast staining. This technique was first introduced by Paul Ehrlich. Later, it was modified by the two German doctors, the bacteriologist, Franz Zeil, and the pathologist, Friedrich Nielsen. So, this method is also popularly well known as Zeil-Nielsen's technique. This is a differential bacterial staining method. Here, more than one stain is used. This technique is used to differentiate and classify the bacterial organisms into two groups: the acid-fast bacteria and the non-acid-fast bacteria. Our interest is to identify the acid-fast bacteria. The very good example of an acid-fast bacterium is Mycobacterium species, which is the causative agent of the tuberculosis, the TB, in humans and animals, and the causative agent of the para-tuberculosis in animals. Certain bacteria, on application of the carbofuchsin staining solution, they can easily take up the stain to their cell surface. Next, on application of the decolorizing agent, the acid alcohol, they can be easily get decolorized, and leaving the bacteria colorless. On contrast, certain bacteria, on application of the carbofuchsin staining solution, they are difficult to stain. But once they take up the stain, further on application of the decolorizing agent, the acid alcohol, they are not decolorized, and leaving the bacteria to stain pink. Such type of bacteria are called as the acid-fast bacteria. The other group of bacteria, which get easily decolorized with acid alcohol, are called as the non-acid-fast bacteria. The very good example of an acid-fast bacterium is Mycobacterium. These mycobacteria are slender rods. The surface of this mycobacterium consists of a lipoidal wall, called as mycolic acid, which is a waxy material, over the mycobacterial surface. So, because of this lipoidal wall, these bacteria, on application of carbofuchsin staining solution, they are difficult to stain. But, along with the continuous heat treatment, they take up the stain by passing through this lipoidal wall. Further, on application of the acid alcohol, the decolorizing agent, they are not decolorized, and leaving the bacteria to stain pink. This is called as the acid fastness. Such type of bacteria are called as the acid-fast bacteria. In general, this acid-fast bacteria cannot be stained easily. However, once stained, it cannot be de-stained. This acid-fast bacteria are, generally, arranged in bundles. This is due to the presence of the waxy material, the mycolic acid, over the mycobacterial surface. Ultimately, this gives a bundle of faggot arrangement. This is the bundle of faggot. The Mycobacterium will have this type of arrangement. So, the arrangement of the acid-fast bacteria is called as bundle of faggot arrangement. Coming to the principle behind the acid-fast staining technique. This technique differentiate the bacteria based on their coloring property. Therefore, we will discuss their coloring property of the acid-fast and the non-acid-fast bacteria in parallel to each other. Once the bacteria are treated with the strong carbofuchsin, which is the primary stain used here, along with the heat treatment, it gives a pink color to both the bacteria. But only the acid-fast bacterium, fixes the primary stain. Next, when the acid alcohol is added, this is the decolorizer. It decolorizes the primary stain easily from the non-acid fast bacteria. But the acid-fast bacteria retains the primary stain. Lastly, on use of the counter stain, the Leffler's methylene blue, that gives blue color to the non-acid fast bacteria and to the background. So, on acid-fast staining, the non-acid fast bacteria in the background appears blue, and the acid-fast bacteria appears pink. This color difference is due to their difference in their cell wall constituents. The surface of the acid-fast bacteria consists of lipoidal wall, called the mycolic acid, which is a waxy material over the mycobacterial surface. But this waxy material is not present over the non-acid fast bacteria. So, on microscopic examination, the background appears blue, and the acid-fast bacteria stains pink. But, on use of the counterstain, the malachite green staining solution, that gives green color to the non-acid fast bacteria and to the background. In that case, on microscopy, the background appears green, and the acid-fast bacteria stains pink. The demonstration. The procedure for the acid-fast staining technique. The materials required are the strong carbylfuchsin staining solution, which is used as a primary stain in this experiment. The acid alcohol, used as a decolorizer. This is prepared by using 3% hydrochloric acid and 95% alcohol. And the Leffler's methylene blue staining solution, which is used as a counterstain in this experiment. These are the three important solutions required for conducting the acid-fast staining technique. Apart from this, the other requirements are the forceps, the blotting paper, the spirit lamp, the distilled water, and the smear, prepared from the sample, which is air-dried and heat-fixed. And lastly, the staining rack. These are the requirements for conducting the acid-fast staining procedure. First, prepare the smear from sample. Spread the smear to the size of about 1 centimeter square, so as to form a thin film. After spreading, allow it to air dry. It may take 2 to 3 minutes for drying. After air drying, fix the smear by heat, by passing over the flame 2 to 3 times with a slow sweeping motion. This is called heat fixation of smear. After the preparation of smear, place the fixed smear on the staining rack. Then, ignite the spirit lamp. First, add the strong carbylfuchsin staining solution over the smear. Cover the entire smear with the staining solution. Heat the stain until vapor just begins to rise. Do not overheat. Allow the heated stain to remain on the slide for 8 minutes. Great care must be taken when heating the carbylfuchsin. Only a small flame should be applied under the slides using an ignited spirit lamp. After 8 minutes, allow the slide to cool down. And then, wash it with the distilled water. Secondly, add the acid alcohol, the decolorizer, over the smear. Cover the entire smear with the acid alcohol. And allow it to act for 2 minutes or until the smear is sufficiently decolorized to pale pink. After 2 minutes, wash it with the distilled water. Lastly, add the Leffler's methylene blue staining solution over the smear. Cover the entire smear with the staining solution. And allow it to stand for 30 seconds to 1 minute. After 30 seconds to 1 minute, wash it with the distilled water. After washing, blot dry the smear using the blotting paper. Now, the stain smear is ready for the microscopic examination. The microscopic examination. Place the stain slide over the stage. Examine the slide under 100x oil immersion, objective microscopy. On microscopic examination. Following the acid-fast staining technique. These acid-fast bacteria are stain pink, which are slender rods, and they are arranged in bundle of faggot arrangement. Due to the methylene blue counterstain, the background is stain blue. This background gives a good contrast for the acid-fast bacteria in pink. So, the result is written as the smear, prepared from the sample, stained using the acid-fast staining technique, is positive for the acid-fast bacteria, with the bundle of faggot arrangement. The example of bacteria is the Mycobacterium species, which possesses this type of microscopic feature on acid-fast staining technique. Coming to the summary of the acid-fast staining technique. First, the preparation of the smear from the sample. After smear preparation, air dry, and heat fix the smear. After fixation, add the strong carbofuchsin staining solution over the smear. Heat the smear with the staining solution for 8 minutes. After 8 minutes, allow the slide to cool down, and then wash it with the distilled water. Next, add the decolorizer, the acid alcohol, over the smear. Allow it to act for 2 minutes, or until the smear is sufficiently decolorized to pale pink. After the smear is sufficiently decolorized, wash it with the distilled water. Lastly, add the counterstain, the Leffler's methylene blue staining solution, over the smear. Allow it to stand for 30 seconds to 1 minute. After 30 seconds to 1 minute, wash it with the distilled water. After washing, blot dry the smear using the blotting paper. Now, the stain smear is ready for the microscopic examination. Place the stain slide over the stage. Examine the slide under 100x oil immersion, objective microscopy. With this, we are coming to the end of the acid-fast staining technique. The detailed protocol for the acid-fast staining technique is available as downloadable link in the below YouTube description. In next video demonstration, we will see about the Leishman staining technique, which is a blood smear staining technique. Hope the lecture is informative and useful. Thank you.